Hello everyone. Welcome to this video where we're going to be talking about horizontal gene transfer. So, what is horizontal gene transfer? It is any transfer of genes between individuals that have no parental linkage. Now, this mainly occurs in bacteria, so these are the organisms I want to talk to you about today. But before we start with the ways that this gene transfer occurs, this horizontal gene transfer, I want to spend a little bit of time kind of refreshing our memory on bacteria. So, as you can see here, I've drawn you a model of a bacteria cell with its main characteristics. The ones I want to highlight to you today are their circular chromosomes. So, their chromosomes are arranged in circles. Unlike us, our chromosomes are arranged in a linear fashion. Bacteria also have small independent pieces of chromosomes. By independent, I mean that they can replicate independently, and these are called plasmids. So they have chromosomes and plasmids that contain DNA. Okay. They also have something called a sexual pili, and this is going to be crucial because it's going to help bacteria connect between themselves. Finally, I want to point out to you that bacterial cells don't have any internal organization. Unlike us, you probably know that we have a nucleus and a nucleolus. Our types of cells are highly compartmentalized, but bacterial cells, it's quite the opposite actually. And this is probably what helps plasmids kind of move around, and we're going to see how that helps us later. All right, so those were bacteria, and I've added small facts for you on the side just in case you want to kind of brush up on your bacteria knowledge. So the first type of horizontal gene transfer I want to talk to you about today is called competence or transformation. And this is essentially absorbing DNA from the environment. So what's actually going to happen? Basically, a bacterial cell is going to receive signals. These are induced by stress or kind of pressure by other bacterial cells. This is something called quorum sensing, where bacterial cells will actually influence each other to behave in a more social-like behavior. So they're all gonna behave in the same way. So they get this signal and they understand that they are supposed to absorb DNA that's kind of floating around in the environment. So this is double-stranded DNA that's floating around in the environment, and all of a sudden, this bacterial cell gets these signals that it needs to absorb this DNA. Okay, so how does it do this? This is where the pili come in, and this is the type 4 pili, and it's going to link to the free-floating DNA. It's going to kind of grab it. One of the strands of this double-stranded DNA is going to pass through the membrane of the bacterial cell, while the other strand of the free DNA, is going to be degraded and used as energy to actually get the first one in. Cool. All right, so now that the DNA has entered into our bacterial cell, what's the point? You're probably going to ask. Well, the point is either going to be to incorporate this new piece of DNA into the bacterial genome, or it can kind of just use the nucleotides of this free piece of DNA as building blocks for its own use. Okay, it's not very important, and maybe at this stage you're not very interested in this, but the way in which the single-stranded DNA is going to be incorporated into the bacterial genome is by a process called homologous recombination. All right, cool. Okay, so now I want to talk to you about the second method, which is called conjugation. Now, there are two subcategories of conjugation, but we're going to see both of them, and this is the first. So, say you have two bacterial cells, and they each have their circular chromosomes. As you can see, I've drawn them in purple here. And they're going to be connected with each other, these two bacterial cells, through their conjugation pilus. So that's that little pink bit that I highlighted in there. Now, say the bacterial cell on the left has this plasmid, what we talked about before, the independent piece of DNA, called the F plasmid. So it has this F plasmid, and we're going to call this cell F plus, positive for the F plasmid, and the one on the right, as you can see, doesn't have it, so we're going to call it F minus. Now what's going to happen is when these two cells come into contact with each other, since there's no internal organization, the genome isn't really kept anywhere, nothing is stopping it pretty much from kind of floating around. So that's pretty much what's going to happen. The plasmid is going to unroll itself, squeeze itself, I guess you could say, through the conjugation pilus, and it's going to poke its head into the other bacterial cell, where the machinery of this bacterial cell on the right is going to start copying it. It's going to start copying it, and by the end, before the two cells have disconnected again, it will probably have copied the entire thing. So at the end, both cells are going to be F plus. They're both going to be positive for the F plasmid. Okay, right. So now I want to look at the second type of conjugation. Now this one is fairly similar to the first, but we're gonna go through it. Same thing as before. You have this bacterial cell. They've got their purple circular chromosome. They've got their F plasmid, so they are an F plus cell. Now here's where it kind of differs. Here, the plasmid is going to get integrated into the genome. We're going to call this cell HFR. So it's going to be HFR F plus. And HFR means high frequency recombination. This basically just has to do with the fact that the plasmid was, through probably homologous recombination, integrated into the genome. So now everything's kind of like in the same place, right? The chromosomes and the plasmid are all in the same entity. Now, same thing as before, the two cells are going to meet, join at their conjugation pilus, and they're going to undergo conjugation. Okay. This new cell, I've drawn the chromosomes in pink. Pretty much same thing happens as before. The donor DNA is gonna get cut, unraveled, squeeze its way in, squeeze its head out to the receiving cell. And this cell is gonna start copying the genome, is gonna start copying this DNA to integrate it, kind of like we saw before, right? Okay. As you can see in the drawing, it wasn't complete. It didn't get completely copied. The cut was done in the middle of the plasmid. So the whole thing didn't get copied and transferred. It only got a bit of the donor chromosome and a bit of the plasmid. So the donor cell, everything goes back to normal, all is good. The receiving cell now has an incomplete F plasmid and a bit of the donor cell chromosome. So it's got this brand new stuff that's coming into him, but the difference with the other one is that none of it is complete. So we can't call this cell F plus because it doesn't have the entire F plasmid. However, what can happen is that these new pieces of DNA get incorporated into the genome, kind of how we saw at the beginning of the diagram, right? It gets incorporated into the genome, and now the genome is made up of part of the plasmid, part of the donor DNA, so the purple one, and his normal DNA, right? The pink one. So we can call this cell HFR for high frequency recombination because it got a bit of DNA and it integrated into its genome. Okay, I hope you see the difference between this type of conjugation and the kind of simpler one that was a bit before. Okay, so the final way of transmitting genes in a horizontal fashion is called transduction. And this one, I have to say, it's my favorite. Only a biology nerd would say this, but I think that this one is really cool. And what makes this one different is that the cells, so the one who gives and the one who receives the genome, never actually come in contact. They never meet. But part of the genome is going to pass from one cell to the other. So how does this go down? Well, this is actually really closely tied with viral infection and viral replication. So if you know a bit about that already, then that is great. Otherwise, we're going to go through the steps one by one. Okay, what happens? You see right underneath the 3 sign, I drew this little guy with a red squiggly line in it. That is our bacteriophage or virus. And this virus or bacteriophage has viral DNA in it, and that's that red squiggly line. So it finds this bacterial cell and it's going to squirt its red viral DNA into it. This viral DNA is very special and it's going to cause the destruction of the bacterial DNA. So as you can see, the blue DNA of the bacteria was circular and it got destroyed, and now it's all these little bits of probably linear DNA because the circle was cut. Okay. The next thing this viral DNA is going to do is going to reproduce itself inside the cell. So there's going to be loads more little red DNA bits. And these red DNA bits are actually going to help form the little guys again. The little viral buddies, I guess we could call them. Not very professional, but it's fine as long as you get the concept. So the next bubble shows these little viral buddies reformed again. And I don't know if you can see from, it's a bit small, but one of them actually accidentally took the blue DNA. It took a bit of the blue DNA that was the bacterial DNA instead of the viral red one. So everybody's going to kind of take all of the viral buddies are going to take a bit of DNA inside of them, like a passport, and this blue fellow, he took the bacterias. But that's okay, because it's not as if the virus has a choice. It doesn't have a mind, right? It kind of just goes with the motions. So this happens. And this little blue viral DNA is going to find another bacteria, a new bacteria cell. As you can see, the new ones are highlighted in blue because the old one, sorry, I forgot to mention this, the old one is going to explode. The old one, the one that got infected, he exploded. It's called lysis of a bacterial cell. This new bacterial cell, which I've highlighted in blue, it's going to actually be attacked by the virus that has the bacteria genome. So this blue genome. And so, as usual, the virus arrives, squirts the DNA into the bacteria. Except here, since it's not viral DNA, it's bacterial DNA, it's not going to do all those nasty things like that it did before, as we saw in the first round. And instead, this bacterial DNA has a high chance of being incorporated into this new cell's genome. So there's a stable gene transfer into the chromosome. As you can see, I drew the dark blue DNA integrating into the chromosome. And that's it. And the two bacteria cells never touched each other. It was all through the, I guess we could say, help of the virus. And those were all of the ways that bacteria transfer their genome between each other. So we saw transduction, we saw two types of conjugation, and we saw competence and transformation. I really hope this was helpful. If you have any questions, I would love to answer them. If you have any other topics that you would like me to cover, I would do that with pleasure, whether it be biology or math or physics or chemistry. I am a nerd for this kind of stuff. So, thank you so much for watching, and I'll hopefully see you next time.